All right, hi everyone. We're going to talk about recombinant DNA technology. So this is another one of the DNA tools, right? And we're going to see a lot of the tools that we spoke about before, like restriction enzymes talked about here. So let's start with, what does recombinant mean, right? Let's look at what does recombinant DNA mean. Well, recombinant DNA is newly generated DNA molecules with DNA from different sources. Recombinant, means you're doing something again, right? Combinant means to combined. So you're recombining different things. You're taking DNA from different places and you're smushing them together into what we call a recombinant DNA. So before we talk about how we get this recombinant DNA and why we might use it and all that stuff, let's look at what recombinant DNA has done. So recombinant DNA technology has revolutionized the way scientists study DNA because it enables us to do what? We can study individual genes. So individual genes can be studied. So I can isolate a specific gene, or you can call it a gene of interest. So you don't forget, these are some words that can be used. So gene of interest. That is not very clear handwriting. Right, gene of interest. So individual genes, specific genes, or in a more biological term, it's gene of interest. Okay? Now, large quantities of this recombinant DNA molecules are needed in order to study them. So we can't just study one recombinant DNA. We need multiple of them. All right, now let's talk about how we make this special DNA. Before we look at what the book has to say, I'll break it down for you. You've got 4 main steps. What you need is, this hasn't been said in your book, but you'll have a donor cell. Basically, this cell has the specific gene that you want to study, the gene of interest. So the first thing we've got to have is our gene of interest. So gene of interest. And then we need something called a plasmid. A plasmid. What is a plasmid? Well, a plasmid is a small, circular, double-stranded DNA molecule that occurs naturally where? Where can we find this DNA molecule? We can find it in bacteria and yeast. Well, what do I need this plasmid for? Obviously, this gene of interest, we need something called a carrier, right? We need something called a carrier, which we call a vector. And what this vector does is it transfers this recombinant DNA into a bacterial cell. And this cell is called the host cell. Now, the reason we want to transfer it to a bacterial cell is because we want it to multiply and go through a process called transformation. But we'll get to that later on. So this plasmid that we find in bacteria and yeast is actually our vector. It's our car. It's our little taxi. It's going to help our gene of interest be transported into our bacterial cell. Right, so that's the first step. We've got our gene of interest and we've got our plasmid. Now what's going to happen is we got to cut those up, right? So we're going to use our restriction enzymes. And the reason we need these is so that we can cut the plasmid and the DNA fragment by the same restriction enzyme. So once you get a DNA fragment, it has a lot of genes in it, right? So you can have, for example, gene A is over here. Gene B takes this area, and etc. So you have this long or short DNA fragment, and you just want gene A. So you're going to get your beautiful DNA scissors that we call restriction enzymes, and they're going to cut this DNA fragment right where your gene is, and you're going to end up with just that red gene, right? Now we're going to do the same thing to our plasmid, and we're going to use the same restriction enzyme. Can you think of why we need to use the same restriction enzyme? Yeah, so that we can cleave them. Cleave means to cut, so that we can cleave them or cut them in the same place, or with this, because what restriction enzymes do is they recognize a certain pattern or a certain code, right? And they cut at that codon sequence, right? So once we cut at those exact same areas for the DNA fragment and for the plasmid, the ends of the DNA will be complementary to the plasmid ends. And then that way we will be able to stick them, right? So if they're complementary, if they have complementary bases because they've been cut at the same area, and we know that restriction enzymes can leave blunt or sticky ends, but if we cut them at the same area, that means they'll have complementary bases. That means I can glue them up together, right? And that's what I need. I want to glue up the plasmid with my DNA fragment so that I can transport it into my bacteria cell. All right, how am I going to glue them? I'm going to use DNA ligase, which is an enzyme, and we talked about this in DNA replication. So what's the DNA ligase going to do? It's our super glue, and it's just going to glue the DNA fragments to the plasmid. All right, cool, we've glued them. Now what? Now, if we look here at the resulting circular DNA molecule that contains the plasmid, which is this blue area right here, and the DNA fragment isolated from the genome, which is the red area, this recombinant DNA plasmid molecule now can be inserted into a host cell so that the large quantities of this type of recombinant DNA can be made. So let's just look really quickly at what happened here. We have the plasmid right here. We cleaved or cut the plasmid with the restriction enzyme. We ended up with these sticky ends, right? And how do I know they're sticky ends? Because they don't have any bases with them, right? They don't have any bases. Here as well, same thing. They don't have any bases paired up with them, so they're sticky ends. And then we got those complementary sticky ends on this side, right? We got those complementary sticky ends on this side. And that helped us to glue up these together into what we call recombinant plasmid DNA. Now that I've got this recombinant plasmid DNA, what am I going to do with it? Now I will insert it into a host cell, and we said the host cell is a bacterial cell, right? So that large quantities of this type of recombinant DNA can be made. So I made my recombinant DNA, now I want larger quantities of them. How am I going to do that? I'm going to insert it into a host cell to create or to replicate more of those recombinant DNA plasmids. And we're going to talk about that in gene cloning and transformation. All right, let's talk about gene cloning. So we made our large quantities of recombinant DNA. We've got those, right? We made our recombinant DNA. Now we need to make large quantities of that recombinant plasmid DNA. So what we're going to do is we're going to transform bacteria. And let's go back to the image here just to explain better. So we have our recombinant DNA plasmid right here. So that's our recombinant plasmid DNA, right? And it's mixed with your gene of interest and your plasmid. And I'm going to explain what an AMP is in a bit once we reach to the end. So what we're going to do is we are going to insert this recombinant DNA into a bacteria. This is our host cell, right? This is what's going to grow or multiply or clone our gene, our recombinant plasmid, right? So that process right here, just highlight that process where the bacteria cell takes up the recombinant plasmid DNA, that process is called transformation. How do we transform bacterial cells? Well, we've got two ways to do it. We can use electric pulsation or heat. How does that work? Well, remember that all cells, including bacteria cells, they have something called a plasma membrane. Now, if you don't know what that is, just go and do a little search about it. But in short, a short electric pulse or a brief rise in temperatures, it can temporarily create an opening in this membrane. Once we have these temporary openings, they allow small molecules like your recombinant plasmid DNA to enter the bacteria cell, right? So as we see here in this image, we see that some bacteria undergo transformation. So some bacteria allow the recombinant plasmid DNA to enter, and some don't, right? So now, once we have this process of transformation complete, the bacteria cell makes copies of the recombinant plasmid DNA during cell replication. So what's different about bacteria and humans is when humans replicate, the genes changes, but when bacteria replicate, they just make clones. What does clones mean? Clones mean exact copies, replicas, right? So what happens during the cell replication of a bacteria is that we create large numbers of identical bacteria. We don't change the DNA, right? There's no change in DNA. It's just copy paste, right? And these identical bacteria each contain the inserted DNA molecule, or recombinant DNA. This process of making the identical bacteria, it's called cloning, right? It's called cloning. Okay, now let's recap a bit here. So we said, some bacteria would undergo the transformation, some won't. That props up the question, well, how do we know if a bacteria underwent the transformation or not? Well, this brings up our friends called antibiotics. So what happens is, we're going to take our recombinant plasmid DNA that contains our gene of interest and contains our plasmid, and we're going to add an antibiotic to it. And the example that the book uses is ampicillin, or AMP, right here, ampicillin. Why would we put an antibiotic? Well, guess what? When you want to, kill bacteria, you're going to put antibiotic on them, right? So if I add to my recombinant plasmid DNA the gene that codes for resistance to antibiotic, the gene that allows to create resistance to antibiotics such as ampicillin, right? That means this bacteria now, if it transformed, if it took my recombinant DNA, that means now it has a shield protecting it from antibiotics because we inserted the gene that codes for the resistance to antibiotics, right? We gave them the shield, okay? So, all right, well, we gave them the shield, blah, blah, blah. What does this mean? How am I going to test it? What we're going to do is we're going to put our bacteria into a Petri dish. And this Petri dish will be full of antibiotics, such as ampicillin. If my bacteria grows on this antibiotic Petri dish, that means it has my shield. And where is my shield contained? Oh, it's contained in my recombinant plasmid DNA. So that means if I have growth, if I have molds that I can see with my eyes, as you can see here, on this Petri dish, right, That means there has been growth. That means the bacteria has replicated. That means the bacteria is living on this Petri dish. That means it is not affected by the antibiotic. It is growing on it. That means it has resistance to it. Whereas the bacteria that has not taken up the recombinant DNA, the bacteria that has not transformed, will not survive on this Petri dish because it has antibiotics, and these antibiotics will kill that bacteria. So the bacteria can't live on it. But the bacteria that has transformed will be able to live and grow in it. They'll be able to survive. So that's how scientists or researchers use this gene that codes for resistance to antibiotics, to distinguish between the bacteria cells that have taken up our plasmid DNA and those that have not taken up our plasmid DNA. And that is the end of the recombinant DNA.